So my pleasure to give you this introduction on PRISMAP. So as an introduction, so we are working with radionuclides which have been and will be used in nuclear medicine and for treatment in Europe and beyond. And from the onset of the discovery of the first radionuclides, for instance, radium, we had the physicists and the chemists at the origin of the separation, let's say, and identification of these radionuclides, which very rapidly understood that the radionuclides would be useful for medical care and more particularly for cancer treatment. And in this line of families and several generations of scientists, physicists and chemists, we had a more recent visit of the granddaughter of Marie Curie, which actually was, is now retired, but was a nuclear physicist and professor from her background. And different radionuclides are, of course, in regular use in diagnosis, and some are emerging now for treatment. And at a high level of institution in Europe or in countries, there is an awareness that there is a need to understand how the supply and the full logistics chains of these radionuclides can take place to not only meet the demand of treatments which are presently in use or in diagnostics, which are presently in use by the medical community, but actually to give the possibility to enable research in this field. If you don't get the radionuclides, then you don't do the research with these radionuclides, and you don't eventually find new properties or new intriguing results which were not expected when working with the most classical ones. And actually in the back, there is a large, not only predicted increase, but actual turnover and economical interest, because if you're bringing in new therapeutics in important fields of medical care, this is translated into important market share and activities for the industry to meet actually, or to provide these treatments when they have passed the proper clearance and clinical testing. And so, as I think a general conclusions or part of this introduction is that indeed the general public then is highly interested to understand a little bit more, because actually many of our colleagues, friends and families do need sometimes or have been exposed to diagnosis, PET CT, scintigraphy and other types of imaging protocol in hospital. So how our starting consortium and PRISMAP program then is aiming at contributing in this field of novel radionuclide. So actually the rationale in the back is indeed to provide and to federate large European infrastructures so that we are able to form this consortium, this network, and then to deliver non-conventional radionuclides. And while this idea of using radionuclides is, of course, then not new, but still, because there is new approaches, there is progresses in detector technologies, in estimation of radiobiological effects of radiations on tissues, and as well even in the production method themselves. Then this program now is expected to, at the European level, then to be able to bring in this field and to produce important results in the coming years. So we are organized, or we are somehow on a very simple few criteria. What is then the, if you want, where are we going with this project, the consortium that we have created, and already the results that we have been producing. So in very simple terms, so we are progressing in the providing the access of these new radionuclides, and this we will see maybe later today, and certainly then in a dedicated working meeting in the rest of the week, on how then new proprietary grades can actually affect and be important in not only using these radionuclides, but on their impact and effect in diagnosis and treatment, and how we can then bring all the information that we will be creating, in this field, on how this can be brought then to a very simple or simplified way and unified way on the, for instance, common entry port and the web interface. And this is very much what we'll be then trying to work on in the coming days. Is how we can then understand the regulatory procedures in the back, and eventually provide more clarity on how we can bring in researchers, scientists in this field of development of radiopharmaceutical, and how then eventually measuring some decay properties, some radiobiological effects, or some production cross-section, how this can then affect that or the other aspect, which then will be involved in the regulatory procedures, and finally how this, when we put this in place, will be then bringing a long-standing structure and consortium for the production of this and making available these new radionuclides. So if we take a very, I would say, rising and specific aspect in medical treatments, there is new radiopharmaceutical, rather new, which are exploiting an emission of an alpha particle. And in this field, there is only a few. It may be looking a lot, but actually we have maybe about 10 radionuclides which may match interesting half-lives and chemical properties and alpha emission, which then can bringing new advantages in treatment for certain medical aspects and disease. And if we take actinium-225, which is actually an isotope which is provided by PRISMAP, the access is not so easy, while there is actually even industrial or partnerships which are getting interest in this field. But up till now, the access to actinium-225 was from sources well identified in US or in Europe. And there is a demand because there is a complex decay chain of this actinium-225, which actually then makes the understanding and the use of this radio element not so straightforward, but which has shown in some clinical testing phases, which is actually superior, if you want, efficacy respect to other radionuclides. So PRISMAP then is a European project which is then making a federation of large-scale facilities which are then providing and producing these radionuclides from different institutes, which is bringing together either accelerators, cyclotrons, or particle accelerators, high energy, high power, nuclear reactors, and mass separation. And I will touch upon a little bit of about mass separation, because I'm in charge of a facility which is making this isotope mass separation at CERN. It's called MEDICIS. But as well, because PRISMAP in its consortium and its spirit then is exploiting advantages that we may find by combining these different methods of production and separation. So PRISMAP is not only, I mean, you will see later if you want the web interface and what we have been working on for the past few months. But actually, then PRISMAP is then structured, if you want, as large projects and large consortium into a way of managing these activities and these services with some steering committee, some coordination team, which is then meeting regularly, and then which is then distributing or channeling in the activities within the different institutions through what we call a work package. And so the framework in which we have today this seminar open to the general public, and then we'll have a working workshop for the subsequent days, actually. So the consortium and some people will be actually working so that we are making progress in this harmonization and standardization of what are properties, what are the regulations that we have to follow, and can we somehow have an emerging understanding on how this can be set to not similar, but at least to coherent or more coherent rules and procedures to follow, so that one radionuclide is leaving the institutes and being used in a laboratory with biomedical researchers. So we are actually then making our best to disseminate, communicate, and train the scientific and even the scientists at large in this field of radionuclides, radiopharmaceutical, and related disciplines. And I wanted to somehow direct you and point you towards our dissemination manager, and as well in charge of the training office, which is Professor Kokolios from Leuven. So all this information you will anyway find either on our web interface or actually through news that you may receive from newsletters, and I will show you how you can subscribe to the newsletter if you're interested in. So the PRISMAP consortium I mentioned, so this is large and renowned institutes which are federated together across Europe, and which is then combining these different, I would say, sophisticated technologies. So it's not, we are not operating a small, if you want, machine or small infrastructures located in a let's say a local institute. So we are really operating important infrastructures in most of the time large laboratories. And the reason why is because the technologies and the rules behind operating, for instance, a nuclear reactor or high power cyclotron requires a very skilled scientist and a very appropriate, I would say, measures of organization of the work and the handling of this radionuclides and these large-scale facilities. We will hear more about these different aspects later with the different presentations we have later today. Why we are interested in having some harmonization on or standardization in the activities we are carrying out in our distributed facilities. And the reason why is because we have research institutes in the consortium which can then welcome, which are used if you want to have proper infrastructures, proper skilled groups which can already do research with this novel compounds or novel techniques exploiting radiopharmaceutical with these non-conventional radionuclides. And the rationale in the back is already in the consortium. We have to make sure that we are properly delivering one radionuclide from one site so that it's purified, integrated in the radiopharmaceutical, and research can be done in an institute part of the consortium. But actually, these radionuclides can be delivered as well to remote places. So if you want to perform research with these novel radionuclides in a certain settings, you actually have to fulfill a number of requirements, licensing, and I would say logical paths, so that you eventually are able to test your compound at the level, for instance, of the clinics, which would be how you qualify a new drug or a new compound and showing a benefit with respect to what was used before. And you see that you have then steps which are then required, and part of this, and not a small part of this, then will be dealing with about showing and demonstrating that you are meeting some standards in the production, the handling, in the toxicity of the compounds you want to handle, and this is sometimes not necessarily following exactly the same path or the same rules, if you want, in one country or the other, already in Europe, but of course in different regions of the world. Always what is in the back is you want to do the activities so that you are protecting people, environment, and you are doing a sound activities so that what you're declaring as the test you want to perform is indeed appropriate with the method you're using for the test you intend to do, and some authorities will be then evaluating whether you're within this framework or not. So the nuclide chart is very big. I mean, you have the periodic table, which is already 100 chemical elements or so. The nuclide chart, you have maybe, let's say, a few thousand radionuclides present, and some or subset of this nuclide chart may be of interest for performing one or other aspects important in nuclear medicine. And this can be either having a treatment isotope or a diagnostic isotope, and I will show why this is eventually important to combine either a diagnostic treatment isotope or different type of treatment isotopes. And then you must comply, as I mentioned, then to a certain number of I would say estimation declaration, and in this we sometimes either rely on properties which were measured maybe long ago and which can be refined, or eventually properties which are not so easy to determine depending on the type of emissions for these radionuclides in certain cases. So the PRISMAP project is expected then to actually bring researchers from across Europe and even groups which had not so much of an idea of what I'm talking about today in terms of licensing, in terms of regulatory aspects, but which simply is probably very competent in one or the other fields which can be developing a technology for imaging, or eventually in radiopharmaceutical or in chemistry has developed a new biological compound which is targeting new biological targets which up to now was not identified for a certain medical case. And so we are, as I mentioned, we have thousands of radionuclides, but some of those do have different chemical properties and emission properties, so that this may bring to an interesting result and an effect which is not necessarily possible, or was not necessarily possible to highlight because you didn't have the proper radionuclides to perform your research. So more concretely, so when I mention a different type of properties or half-lives, or even type of purity grade that you may want to exploit and make available to research groups across Europe. If we take a case of lutetium-177, already, which is now a marketed compound for some treatment of neuroendocrine cancers, already there is actually an impact of the grade you will have when you produce this lutetium-177 on possibly the outcome of using this simply to check for therapeutic effect, but even for waste management or eventually patient care, if this patient can go back home or not in a certain conditions. And this depends very much on the type of emission, the type of, as I mentioned, purities that you can exploit. You have more, let's say, illustration of what we can, how we can bring this development at the level of the large-scale facilities with new ideas and new direction you can take in research in this field of radiopharmaceutical with the non-conventional radionuclides. And here this is just let's say some very subjective selection of how you can see different directions that you may or we may be taking in Europe in the coming years. And you will see from techniques to demonstration of, let's say, principal demonstration of some of the radiopharmaceutical which are used for the first time in preclinical research, how this can be providing interesting results. But you see already how we are asking ourselves on even if you get proof of principle at a certain level, how can you then streamline that later for research into the clinics? We know the path is long, and so this is important then to promote and highlight these channels. And to give you a concrete example, that with a single chemical element, which is terbium, you can revary either for treatment as you would have with more classical radionuclides which were available, but the fact that you can combine an imaging capacity with the same radiopharmaceutical, we call that as a really matched pair or matched combinations of elements, because you are able to then make the imaging together with the treatment. Of course, the regulation will not be the same for one or the other radiopharmaceutical, but certainly one is not excluding the other. And then we are now in this field of theranostics, combining these diagnostics and therapeutic effect, whereby the combination itself you may actually bring advantages with respect to what was done eventually before. So the projects are expected to come in PRISMAP through actually call for projects. So there is an open call now. You can go on the prismap.eu website and have more understanding on how the researchers, even if not directly in the field already, may apply and may submit a competitive research, which then will be evaluated. And this is, for instance, how it has been published or advertised in this European Association of Nuclear Medicines already. And to give you a bit of maybe technical aspects, more on what we are working on now, even at the level of production, and then future qualification of this radionuclide. So you have Isolde and Medicis, which is then part of the CERN accelerator complex, which otherwise is a high energy physics leading world institutes for high energy physics, with, for instance, you may know more the discovery of Higgs. And you see that we are producing all kind of particles, and in our case, what we're interested in is the production of radioactive ion beams. And you see this is part of the many particles which is accelerated throughout the CERN complex. And why this is of interest even in this framework, or in the coming days, where we have started and triggered the understanding of standardization qualification of radionuclides, is because the process of mass separation goes just beyond what was already well accepted and well developed for the field of medical radionuclides, with production either in reactors or cyclotrons, with important aspects like what is the purity of the target you will be using to produce this radionuclide, what will be the cross-section, what will be the irradiation parameters, etc. In the case of mass separation, then in addition, we have the process, if you want, of the separation itself. So the radionuclides are not only produced through nuclear reactions in the target itself, but then are mass separated from the target by, if you want, distillation and ionization. And this process is specific in itself, is new in this field of medical radionuclides, at least for radionuclides to be brought to the clinics. And some of the possible expectation, and I'm soon to the end, of the expectation is that eventually, if you are able to separate out radionuclides from the starting target, not by radiochemical way, but by this physical mass separation, you may have interesting high specific activity. And this is certainly a parameter which was already, let's say, identified as being able to bring different efficacies for a certain compounds, but which then is probably brought at a new level now with this PRISMAP consortium to really match some efficacy, because you can accumulate now this targeting ligand to targets identified on the cancer cell, which then would enhance the contrast of either the imaging or the treatment by proper targeting, proper stability, proper eventually translocation of your radionuclides in the tissues you're targeting. So I'm very close to the end. So you see that we are putting a structure in Europe to really address what is in the back to what ultimately led to some companies to support and validate at the highest clinical requirement the use of a certain radiopharmaceutical for treatment at the end, eventually for research already with patients to demonstrate their clinical efficacy. This is then our ambition to eventually bring new radionuclides or non-radionuclides which were not possibly used in certain combination of diagnostic and treatment, which now will eventually become possible. And this is by bringing these research groups to this PRISMAP platform. So for my concluding note, so I hope you will then enjoy this seminar today, and we will hear more about how we produce, how we what we are looking at for having the possibility to manipulate these radionuclides, eventually test them in proper settings. If you are interested to and not part of PRISMAP, you should definitely use and join our user forum and get newsletters. You can get it already from prismap.eu, but if you want to get regular updates, do subscribe on our user forum. And this first day and actually subsequent days for us is an important meeting in less than a year of existence of PRISMAP, which is organized then in hybrid mode, right, in Innsbruck, where we will then set, if you want, the first steps towards standardization and understanding of the full supply chain, if you want, and characteristics that we need to fulfill from the production to the use and applications for research in this field. So that was the concluding remarks. So thank you very much, and yeah, looking forward for the rest of the presentations. Thank you.